Somebody confirm you can hear me?
Yeah, we can hear you.
Great. Thank you. Let me get to chapter 5. Before we begin, are there any questions? Let me share my screen. Should be seeing my screen now. Today is October 29th, and we should be starting. No, we shouldn't be starting chapter 5. We should be midway through chapter 5. Um, Let me remind you there's going to be a quiz this Wednesday, quiz 3. Quiz 3 will cover labs 5 and 6, chapter 4, and the end of chapter 3. Any questions about any of that? In the lab today, we'll do 11. Let me look to see. All right, I do have a mark. So I do remember talking about photosynthesis. Photophosphorylation only occurs in photosynthetic organisms such as green plants and green bacteria. And in photophosphorylation, this is one of the three ways that cells can make ATP. The other two ways are by respiration and then by substrate level phosphorylation, where you move a phosphate from one molecule onto ADP. In metabolic pathways for energy production, you can generate energy from the energy in molecules like food, and it's not released in a single burst. It's released in a series of reactions, and I kind of showed you that already. In catabolism, you can remove the energy from a food molecule such as glucose, and then it goes through a electron transport chain, carrying the electron from one molecule to the next. So the energy is given off in increments. At each passage of the electron here, energy is given off. And along the way, ATP can be generated. We'll talk about that when we talk about aerobic respiration. So electrons from donor molecules are passed from one compound to another through a series of redox reactions, and they're giving up their energy in slight increments. And that's why a cell can make ATP from burning glucose and stay at around 37 degrees. Because if you were to light the glucose, you would be something like 1,000 degrees. I don't remember if that's Fahrenheit or Celsius, but it's real high temperature. So in carbohydrate catabolisms, most organisms can use glucose to obtain their cellular energy. And most organisms can engage in carbohydrate catabolism to make ATP. The most common energy source is glucose, but organisms can use other molecules, such as complex carbohydrates and lipids and proteins. Even humans can digest lipids and proteins, as well as obviously complex carbohydrates. But for those of us couch potatoes, we tend to convert all of these molecules into glucose or some other sugar and then send it through aerobic respiration in the normal way. You can burn lipids, like a marathon runner can directly burn lipids to generate energy, meaning to make ATP and then use the ATP for their muscle movements. They can use lipids, but a marathon runner does not burn lipids until 45 minutes into their marathon run. Any question about any of that? So initially, a marathon runner is making ATP, or making their own energy, from burning glucose or another carbohydrate, in which case the carbohydrate is broken down into glucose. That isn't always true. Like, for example, fructose is not easily broken down into glucose, and you have to take the fructose, digest it, take it into the bloodstream, take it to the liver, and then the liver will slowly convert it to glucose. But all other carbohydrates will be easily changed into glucose and then released into the blood as glucose. Any question about any of that? All right, to produce energy from glucose, microorganisms use two general processes. They can use respiration and they can use fermentation. Now, respiration has two forms, aerobic respiration and anaerobic respiration. And in this lesson, we'll talk about both of them, primarily focusing on aerobic respiration. Any question about any of that? If there's no questions, let's watch a little video introducing aerobic respiration, assuming that it gets started here. It's taking its time. And we have to watch a commercial first, which is also.
Once everything's perfectly organized, I'll start working on my thesis.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
All right, any questions about any of that? So let's talk more about aerobic respiration, and we will compare it to fermentation. We will talk about fermentation after we've talked about respiration. Aerobic respiration has three principal stages. There is glycolysis, the Krebs cycle, and then the electron transport chain. Essentially, electrons flow from glucose, an energy-rich molecule, to CO2 and water, which are energy poor molecules. Aerobic respiration has a high yield of ATP. Comparing to fermentation, fermentation does have glycolysis, but no Krebs cycle and no electron transport chain. ETC stands for electron transport chain. Instead, pyruvate is converted to a product, the product of fermentation, and it depends on which fermentation reaction we're talking about. We will talk about ethanol and lactate or lactic acid being made. In fermentation, you have a much lower ATP yield. This slide is only showing you a summary of aerobic respiration compared to fermentation. But in both processes, we can use glucose, shown here, to make ATP. Any question about any of that? All right, let's talk more about aerobic respiration. Once again, we're showing you an overview. This is showing you aerobic respiration in a eukaryotic cell. Aerobic respiration has three parts: glycolysis, breaking down glucose into pyruvate. Glycolysis occurs in the cytoplasm of the cell. So if this pink box, let me blow this up a little. If this pink box were to be a cell, the pink would be the cytoplasm. And then in glycolysis, we make NADH. I'll talk about that a little bit later. We do make a little bit of ATP, and then we make pyruvate from glucose. The pyruvate then leaves the cytoplasm and in a eukaryote enters the mitochondria. Once that happens, the Krebs cycle occurs, and it's actually the preparatory step in the Krebs cycle. We'll talk about that in a little bit. The Krebs cycle has different names, including the citric acid cycle. I and your textbook tend to call it the Krebs cycle after the doctor who discovered it, Dr. Krebs. It does have more names. I'm trying to remember what the other ones are. It's also called the citric acid cycle and the tricarboxylic acid cycle, or the TCA cycle. You don't need to know the other names, but you should know the Krebs cycle. And I won't test you on what's another name for the Krebs cycle. In the Krebs cycle, we do make a little bit of ATP. We do make some CO2. And more NADH and FADH2 are made. NADH and NADH from both the glycolysis and the Krebs cycle, as well as FADH2 from the Krebs cycle, are electron carriers carrying electrons from glucose to the third part of the aerobic respiration, the electron transport chain, which is shown here as electron transport. Electron transport has different names, including oxidative phosphorylation, chemiosmosis, and it's called electron transport chain or electron transport phosphorylation. Electron transport phosphorylation and electron transport chain are similar enough that doesn't tend to confuse students. I don't use the term oxidative phosphorylation or chemiosmosis just because these terms are so different. But I do tend to use electron transport chain, which is the term that your textbook uses, and the abbreviation for that is ETC. In the electron transport chain, the electrons from glucose in the glycolysis or from the derivative of glucose in the Krebs cycle are carried to the electron transport chain to generate ATP. Now, the citric acid cycle in a eukaryote happens inside the matrix of the mitochondria. The electron transport chain happens across the inner mitochondrial membrane. Any question about any of that? Now that is for a eukaryote cell. In a prokaryote cell, we'll talk about that a little bit later. They don't have a mitochondria. And what they're doing is glycolysis happens in the cytoplasm, which is normal. And then the citric acid cycle and the electron transport chain happens near or across the cell membrane. Go down a little bit. So in aerobic respiration, the first step is glycolysis. Glycolysis does have another name. I don't use it. I think your textbook uses it one time just to mention it. And it's the Emden-Meyerhof pathway. And I'm not German, so I could be butchering their names. This is where the oxidation of glucose occurs, making pyruvic acid. Glycolysis occurs in most living cells, and it can occur in the presence or in the absence of oxygen. Glycolysis does not use oxygen. It does produce ATP and NADH, but only a little bit of ATP and a little bit of NADH as well. What do I want to talk about that? Oh, here it is here. Pyruvic acid is sometimes called pyruvate. The cell uses pyruvate and pyruvic acid the same. You would need a chemist really to distinguish the two. Pyruvic acid has the hydrogen attached to it. Pyruvate does not. And in glycolysis, you convert NAD plus to NADH. And NADH acts as a carrier of electrons. We talked about electron carriers in our last lesson. And here we're talking about NADH. Any question about any of that? All right, this is showing you glycolysis, the 9 or 10 steps in glycolysis, where, let me blow this up if I can. Glucose, which is a 6-carbon molecule, is converted into different steps, resulting in a 3-carbon molecule called pyruvic acid or pyruvate. And along the way down here, you do make 2 3-carbon molecules, but this one here can be converted into that one. And so eventually we end up with 2 pyruvates from 1 glucose. And that's because a 6-carbon molecule makes 2 3-carbon molecules. Any question about that? Along the way, here's we're making the NADH, and then we're making ATP, 2 of them, and 2 of them here. But to get glycolysis to run, you do have to put in 2 ATP. So our net gain of ATP from glycolysis is plus 2. Any question about any of that? Now, when I was an undergraduate, I needed to know the names of all of the intermediates, which are given here, as well as the enzymes to make each of the intermediates. And since I was required to know that, I think it's fair that you guys are required to know that. What do you guys think?
We can do it, probably.
I'm not going to ask you to memorize the different intermediates of glycolysis. Just know the summary reaction, which is shown in this slide, that you start with glucose, you get a net gain from glycolysis of 2 ATP, 2 pyruvates, and you could call those pyruvic acids, and 2 NADH. And there's the summary reaction, which you don't need to know, where glucose 2 ATP, 4 ADPH, 4 inorganic phosphates, 2 NAD pluses generate 2 pyruvic acids, 4 ATP, 2 NADH, 2 hydrogen, 2 water. You subtract 2 ATP from 2 ATP, and you get a net gain of 2 ATP from glycolysis. So you must know what's in the red box for glycolysis. Any question about any of that? The next step in aerobic respiration is glycolysis excuse me, not glycolysis, but the preparatory step in the Krebs cycle. Before I talk about the next step in aerobic respiration, let me inform you that there are 2 forms of respiration. Aerobic respiration, which we're going to talk about now in detail, and anaerobic respiration, which we'll talk about a little bit, not a lot, but we will talk about it, comparing it to aerobic respiration later, after we talk about aerobic respiration. Respiration is the oxidation of molecules that releases electrons for an electron transport chain. So as we've stated, the electrons flow from glucose and they're carried by NADH or NADH and FADH2 in the Krebs cycle to the electron transport chain. In aerobic respiration, the final electron acceptor is oxygen. So oxygen is the molecule that the electrons which flow from glucose end up in. So oxygen is the final electron acceptor in aerobic respiration. In anaerobic respiration, the final electron acceptor is a molecule other than oxygen. It depends on which form of anaerobic respiration we're talking about, what the final electron acceptor is. It can be iron, it can be nitrate, it can be nitrite, it can be sulfate, and there's different forms of anaerobic respiration, and each of them have a different final electron acceptor. Along the way in respiration, ATP is generated by oxidative phosphorylation, and that happens in the electron transport chain. There is some ATP made in the Krebs cycle, but only 2, and we'll talk about that in a little bit. So you can make ATP a little bit in glycolysis, and you can make a little bit in the Krebs cycle, shown here, but the majority of ATP made in aerobic respiration happens in the electron transport chain, and that's the 3rd step of aerobic respiration. Any question about any of that? All right, so pyruvate comes out of glycolysis and then moves into the mitochondria. The pyruvate cannot join the Krebs cycle directly, so it's changed in the preparatory step from pyruvate or pyruvic acid to acetyl coenzyme A. Along the way, you do generate some NADH, 1 for each pyruvic acid, and 1 CO2 also for each pyruvic acid, meaning the pyruvic acid is a 3-carbon molecule. Hopefully you can see that. And acetyl coenzyme A is a 2-carbon molecule. So 1 carbon comes off, and it's comes off or burnt as CO2. And this happens in the preparatory step. Now, for each molecule of glucose, you generate 2 pyruvic acid molecules. So the preparatory step runs 2 times for each molecule of glucose. Any question about any of that? All right. The preparatory step then goes to the Krebs cycle, meaning acetyl coenzyme A can directly enter the Krebs cycle. The Krebs cycle, as I mentioned before, has different names. The tricarboxylic acid cycle, or TCA for tri-carboxylic, C for carboxylic, and then A for acid. It's also called the citric acid cycle, T for the, C for citric, and A for acid. Any question about any of that? I won't use these other names. I'll always use the term Krebs cycle. And generally, your textbook also calls it the Krebs cycle. In the preparatory step and the Krebs cycle, meaning the preparatory step is shown here, converting pyruvic acid into acetyl coenzyme A, we do generate one ATP that happens here in the Krebs cycle. You do generate 4 NADH, one in the preparatory step here, one right here, one right here, and then the fourth one right there. And then you generate one FADH2 molecule right here. And then you generate 3 CO2 from one pyruvic acid. There's one CO2 here in the preparatory step, one here in the Krebs cycle, and one there in the Krebs cycle. So all of the carbon from pyruvic acid is converted into CO2. And that is why we breathe out CO2, because we convert glucose in aerobic respiration into CO2 and into water. I haven't shown that yet, but the glucose is converted into CO2 and water. And here we're generating all of the CO2, and we need to breathe that out. None of that CO2 that we breathe out comes from oxygen. All of the CO2 comes from a carbon in pyruvic acid, which you remember in glycolysis, the pyruvic acid comes from glucose. And so you must know in the preparatory step in the Krebs cycle, this box shown in red, that you generate 1 ATP, 4 NADH, 1 FADH2, and 3 CO2. However, for glucose, the preparatory step in the Krebs cycle runs 2 times because glucose makes 2 pyruvic acid molecules. So the preparatory step in the Krebs cycle run 2 times. And so if I ask you a question, how many NADH are generated by the preparatory step in the Krebs cycle, you tell me 4 NADH per pyruvic acid. However, if I ask you how many NADH are generated by glucose from the preparatory step in the Krebs cycle, you have to take this number and multiply by 2, or 8 NADH are generated from a molecule of glucose. Any question about any of that? All right, I've already mentioned the NADH and the FADH2 as well as the H+, which is generated. It doesn't show on here. It's generated in one of these places with NADH being made. These are destined to go to the electron transport chain, the ETC. And then notice this Krebs cycle. Let me see if I can blow that up a little. It starts with acetyl coenzyme A joining with oxaloacetic acid to make citric acid. You don't need to know the citric acid, which is converted to this molecule, converted to this molecule, converted to that molecule, converted to that molecule, and we make ATP here, converted to that molecule, converted to this molecule, and then converted to oxaloacetic acid, And that's the molecule we started with. So the Krebs cycle is a cycle that will continue in your cells for as long as your cell is alive. And you have something to burn like glucose. Obviously, if you don't have any glucose around, the Krebs cycle won't occur. But if you have a molecule of glucose or something that can be converted into glucose, the Krebs cycle will always be going on in your cell. Any question about that? And that's why it's called a cycle. It isn't a one-way pathway that ends. It will be a cycle that continues in a circle. Any question about any of that?
I do have a quick question.
Huh?
For the hydrogen ion, is that kind of just making its way to the next stage, the electron transport chain, or is that like does NADH also include that hydrogen ion in it? Like, does it need a carrier? Is what I'm trying to
No.
say.
Remember I mentioned that the hydrogen ions tend to follow the electrons in um, bios biological reactions. Well, here we have actually two hydrogen ions. Come on, mouse. My mouse died for some reason. Oh, there it is. Because we start with NAD plus, and it's converted to NADH. So one hydrogen ion and two electrons join NAD plus. And then another hydrogen ion follows the NADH to the electron transport chain. And you don't really need to know about the hydrogen ion because it's not carrying an electron. Um, you do need to know about NADH because it's carrying an electron and it's also carrying a hydrogen ion. Okay. Uh,
Gotcha. Yeah, thank you.
so let's just say that the hydrogen ion will follow the NADH, and the NADH is itself carrying a hydrogen ion as well as two electrons. And the important part is the NADH is carrying the two electrons because the electrons are going to drive the electron transport chain. So NADH is called an electron carrier. The H plus are kind of important, but not as important as electrons. And we'll talk about that in the electron transport chain. Okay?
Cool. Thanks.
All right. So this is once again showing you the overview of respiration compared to fermentation. And we're going to change that to aerobic respiration because that's what I'm talking about. Although anaerobic respiration happens to be the same summary. And that is we had glycolysis making some NADH and some ATP. And then we go to the preparatory step in the Krebs cycle, making some ATP and a lot of NADH, as well as FADH2, which isn't shown here. And then NADH from both glycolysis and the Krebs cycle, and then FADH2 carry the electrons that originally came from glucose to the electron transport chain. And that's what this summary slide is trying to show you. In the electron transport chain, we'll then use these electrons to generate ATP. And that's just comparing it to fermentation, which is making some ATP in glycolysis. And then we actually regenerate the NADH, we'll talk about that, to make a fermentation reaction. And then you have the end product of fermentation, which we will talk about two of them. Lactic acid fermentation, the end product is lactic acid. And then ethanolic or ethanol fermentation is makes ethanol. Any question about this overview slide? All right. So we now come to the electron transport chain. And at the moment, we're only talking about aerobic respiration. Although in anaerobic respiration, we do have the electron transport chain as well. The electron transport chain is a series of carrier molecules, mainly proteins, that are in turn reduced and oxidized as electrons, which initially came from glucose, are passed down the chain. Energy is released as the electrons are transferred from a high energy compound to the next step, which is a lower energy compound. Energy is released and can be used by chemiosmosis, or part of the electron transport chain, to produce ATP. And let's watch this little video introducing aerobic respiration. No commercial this time.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase.
You don't need to know those
In
names.
addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C.
You don't need to know any of these.
Other
That's
key
the last one.
components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Any questions about that video? All right, let's take a look at the electron transport chain. In aerobic respiration, the electron transport chain occurs along and across the inner mitochondrial membrane in a eukaryote cell. Prokaryotes do not have a mitochondria, And so the electron transport chain occurs along and through the cell membrane. So here we have a membrane, and this is called an electron transport chain because the electrons from NADH and FADH2 are given to the proteins in this membrane, and then they're transferred to one protein or carrier to the next. So the electron is transport in a chain. And in the eukaryote, this membrane is the inner mitochondrial membrane containing all of these proteins and carrier molecules. In a prokaryote, this membrane is the cell membrane. Any questions about any of that? All right. When NADH comes up to the electron transport chain, it gives off a hydrogen ion and 2 electrons to the first protein in the chain. As these electrons are moving through that protein, we give the protein the energy it needs to transfer a hydrogen ion from this side of the membrane to that side of the membrane. The electrons are then transferred to this protein and then transferred to this protein. And this protein, once again, when the electrons move through it, it gives the protein the energy it needs to transfer 1 hydrogen ion from this side of the membrane. Come on, mouse. To that side of the membrane. And then the electrons move to this carrier or protein in the membrane, to this carrier or protein. This is a protein actually in the membrane. And this protein, as the electrons move through the protein, it gives the protein enough energy to transfer 1 hydrogen ion from this side of the membrane to that side of the membrane. So within ADH, we're actually getting 3 hydrogen ions pumped from this side of the membrane to that side of the membrane in the electron transport chain. But for FADH2, we only get 2 hydrogen ions moved from this side of the membrane to that side of the membrane. And that's because FADH2 starts lower in the chain than NADH. And so it's skipping the first protein, which does transfer a hydrogen ion to the side of the membrane. So I guess you can say NADH is more efficient than FADH2. And you can get more ATP generated from NADH than FADH2. Any question about any of that? All right. So we have a bunch of hydrogen ions which are pumped from this side of the membrane to that side of the membrane. That creates a potential energy difference between this side of the membrane and that side of the membrane. This potential energy is from the hydrogen ions wanting to move from this side of the membrane to that side of the membrane. And the gradient is twofold. There's more hydrogen on this side than that side. And so hydrogen will want to move from areas of high concentration, meaning this side. Come on, mouse. This side to that side. Excuse me. As well as there's more positive charges on this side of the membrane than on that side. So these positive charges will want to move to this side of the membrane. This potential energy difference is used by this protein to make ATP. And that is when hydrogen ion, it can only move across the membrane at this protein. Come on, mouse. And when the hydrogen ion moves through this protein, it gives the protein the energy it needs to add a phosphate group to ADP to make ATP. So this molecule is called ATP synthase. Any question about any of that? Now, how efficient is this in generating ATP? For one glucose molecule, about 40% of the energy in glucose, will be converted to the energy of ATP. And you might say, well, that's not very efficient. That's less than 50%. However, if you talk about your car engine converting the energy in gasoline into the energy of the car moving, do you know how efficient we have made the best motors we can make? Anyone have a guess? Anyone actually know?
30%?
That's a good guess. You're really close. It's the very best motor they can make can convert the energy in gasoline only about 25% to the energy of the motor, meaning the car moving. And that would be a totally experimental motor and car, something made out of plastic, and you wouldn't be able to drive with it because it would just fall apart if you went over a pothole or got in an accident. And that's the very best we can do, 25%. So your cells are much more efficient. And actually, aerobic respiration is the most efficient way of generating ATP. Any question about any of that? All right. Along the way, the electrons, which come from NADH or FADH2, move through these proteins. They're transferred from one protein to another in a chain, which is called the electron transport chain. The electron will come to this last protein and then transfer the electrons to water. Excuse me, to oxygen, not to water, to oxygen. And that's why it's called aerobic respiration, because the electron, the final electron acceptor, is oxygen. Let me see if I can blow that up a little. The electrons leave this molecule and transfer two electrons, and oxygen plus two electrons and plus two hydrogen ions form water. And this is actually responsible for all of the oxygen you breathe in. All of that oxygen is converted into water in aerobic respiration. None of that oxygen is converted into CO2. In us, some of that water is converted, and then you can use it for water, for your water needs, your drinking needs, or whatever. Humans aren't very good at doing that, but if we're talking about desert animals, they can convert the water from aerobic respiration into the water that they need, and they can get 65% of the water they need from aerobic respiration. Humans aren't very good at keeping this water, and so we breathe out a lot of this water. We do keep some, but we probably breathe out more than 50%. I don't know what we breathe out, but we're not good at keeping the water. And that's how we generate water in aerobic respiration. The electrons and the hydrogen ions combine with oxygen to make water in the electron transport chain. Any question about any of that? So in the electron transport chain of aerobic respiration, we generate between 32 and 34 ATP per molecule of glucose. Prokaryotes are a little more efficient, and they generate 34 molecules of ATP per molecule of glucose. And like I said, an electron transport chain of aerobic respiration, the final electron acceptor is oxygen. So the electrons end up at oxygen. which then combines with hydrogen to form water. Any question about any of that? So this is showing you the electron transport chain in greater detail. Let me go to the next slide. This is showing you, well, let's go back here. Glycolysis right here. And then the Krebs cycle and the preparatory step, which isn't quite shown, it's before this. The Krebs cycle. The NADH and FADH2 come from the Krebs cycle. The electrons being transferred from one carrier molecule to the next happen in the electron transport chain. And then the electrons flow to oxygen to generate water. I'm showing you once again, let me see if I can blow this slide up. In aerobic respiration of a eukaryote, this membrane, which is where the electron transport chain occurs, is in the inner mitochondrial membrane of the cell. And so these hydrogen ions are being pumped in between the inner membrane and the outer membrane of the mitochondria. And in eukaryotes, this is really good. These hydrogen ions can't damage the cell because they're kept inside the outer membrane of the mitochondria. Any question about any of that? Now, in a prokaryote, they don't have a mitochondria, so this electron transport chain is happening right here, and that's the cell membrane of the cell. And so where are these hydrogen ions being pumped in a prokaryote cell? It's not between the inner and the outer membrane because there is no, outer membrane of a mitochondria. Where is the hydrogen ions being pumped in a prokaryote cell? Well, where's the cell membrane? It's right here, right? So what's outside of the cell membrane in a prokaryote? Come on, people. What's outside the cell membrane in a prokaryote cell? Somebody said something. I didn't hear it.
I just said the paraplasmic space.
It's pumped into the cell wall. So if the cell has a cell wall, and most bacteria have a cell wall. I think most archaeobacteria also have a cell wall. I'm not an expert on the archaeobacteria, but I think most do. So in most prokaryotes, these hydrogen ions are actually pumped in the cell wall. And yeah, if it's a gram-negative cell, it will be in the paraplasmic space, meaning the outer membrane will actually keep the hydrogen ions inside the cell wall. In a gram-positive cell, though, these hydrogen ions could leave the cell wall and then leave the cell, but they do want to move to this side of the membrane, so they will stay near the cell wall and come to the ATP synthase, and then move through the ATP synthase protein to generate ATP. The point is, in a prokaryote cell, these hydrogen ions are pumped into the cell wall, if the cell has a cell wall. And most bacteria do. But in a eukaryote, it's pumped into the space between the inner mitochondrial membrane and the outer mitochondrial membrane. The pumping of the proteins making the gradient of hydrogen ions is called the proton motive force. I won't quiz you on that, but your book does talk about it. And I did talk about the gradient of hydrogen ion. Chemiosmosis is the term for the movement of hydrogen ion from this side of the membrane to that side of the membrane, making ATP. And the electron transport chain is also called oxidative phosphorylation because ADP is phosphorylated, and it's aerobic respiration, why it's called oxidative. But I don't use the term oxidative phosphorylation. I'll just call it part of the electron transport chain. So the complete oxidation of glucose via aerobic respiration, including glycolysis, the preparatory step, and the Krebs cycle, and the electron transport chain, generate 36 to 38 ATP molecules per molecule of glucose in a eukaryote cell. In a prokaryote, they're a little more efficient, and aerobic respiration generates in glycolysis, the preparatory step in the Krebs cycle, and the electron transport chain, 38 ATP molecules. Let me briefly talk about eukaryotes, and that is most of the cells of your body can only generate 36 ATP molecules per molecule of glucose. However, the cells in your heart, your liver, and your kidney can generate 38 molecules of ATP per molecule of glucose. And why do you think your heart, your liver, and your kidneys are more efficient than all the other cells in your body? And you probably don't know about your liver and the kidney, but you should know, what is the heart always doing? Until you die, what's your heart always doing?
Pumping blood.
Yeah, it's always working, it's always pumping. And so the heart needs a lot of ATP to always work. And so the heart, the liver, and the kidneys are always working. And I'm telling you now, the heart, the liver, and the kidneys are always working. And so these cells are a little more efficient at generating ATP. And they can generate 38 ATP molecules per molecule of glucose. The rest of your cells, like your skin cells, the cells of your nose, your blood cells, your brain cells, are not always working. And so they're not as efficient. And they generate 36 ATP molecules per molecule of glucose. And I'm not being totally facetious when I'm telling you your brain cell is not always working. Your brain cell doesn't always work. Most of the cells in your body do not always work. And so they're not as energy efficient. And you don't need to know how the ATP is more efficient in the kidney, the heart, and the liver cells. I'll briefly mention it, and that is in glycolysis. Let me see if I can go back and show that. Oh, I can actually show it here in this slide. In glycolysis, these NADH molecules in the cells coming, or the NADH from glycolysis coming from all the cells except for the heart, the kidneys, and the liver, these NADH molecules are converted to FADH2 in the mitochondria. But in the heart, the liver, and the kidneys, the NADH molecules stay as NADH, and so they're more efficient, the heart, the liver, and the kidneys, at generating ATP. Any question about any of that? All right, you need to know the summary equation for aerobic respiration, and that is you get glucose, C6H12O6, 6 oxygen molecules, 38 ADP molecules, and 38 inorganic phosphate molecules combine to make 6 CO2. All of these carbon come from the glucose, carbon and glucose, and 6 water molecules. All of these come from oxygen, and then 38 ATP molecules. And that's the overall summary equation for aerobic respiration. And this is once again showing you a comparison between aerobic respiration and fermentation. No, this isn't showing you fermentation. This is showing you the making of ATP in glycolysis, the making of ATP in the preparatory step in the Krebs cycle, and the making of ATP in the electron transport chain. Let me blow that up so I can read it. And this is showing you for the heart, the liver, and the kidney cells because you're only making 38, or for a prokaryote cell because you're making 38 ATP. Any question about any of that? All right, let's briefly talk about anaerobic respiration. Anaerobic respiration is a form of respiration where the final electron acceptor is not oxygen. It is a molecule other than oxygen, which is accepting the electrons that ultimately came from glucose. It's an inorganic molecule, and it will differ depending on which form of anaerobic respiration we're talking about. Sometimes it can flow to nitrate ions, other times to nitrite ions, sometimes to sulfate ions, and sometimes to ions of iron. Anaerobic respiration usually has glycolysis, the Krebs cycle, and the electron transport chain. However, the electron transport chain for anaerobic respiration is shorter than for aerobic respiration. And so the amount of ATP you make for the electron transport chain of anaerobic respiration will be less ATP that's made in aerobic respiration. The ATP yield for anaerobic respiration is something greater than 2 up to 36 molecules of ATP per anaerobic respiration. And the exact amount of ATP will depend on which form of anaerobic respiration we're talking about. Some of them are more efficient, and the most efficient you can get is 36 molecules of ATP per molecule of glucose for anaerobic respiration. The least efficient is something like 4 molecules of ATP per molecule of glucose. It's always greater than 2 and 36 or less than 36. Therefore, anaerobes, particularly prokaryotes, generate less ATP in anaerobic respiration than aerobes. And therefore, anaerobes tend to grow more slowly than an aerobe, because aerobic respiration is the most efficient method there is for generating ATP. Any question about any of that? So this is showing you in respiration, generally, both anaerobic and aerobic respiration, we have glycolysis, we have the Krebs cycle and the preparatory step, and the Krebs cycle. I should be saying it in its order. And both of them make NADH and some ATP. And then that NADH or NADH and FADH2 flow to the electron transport chain. This electron transport chain is smaller And so we get less ATP being made by anaerobic respiration, meaning there's less proteins in the chain of proteins in the electron transport chain. Along the way, you do get ATP from the electron transport chain, and the electrons ultimately coming from glucose flow to a final electron acceptor, which in the case of aerobic respiration is oxygen. In the case of anaerobic respiration is a molecule other than oxygen. All right, let's talk briefly about fermentation. In fermentation, you can generate ATP, but you do not gain much ATP. Fermentation releases energy from oxidation of an organic molecule such as glucose via glycolysis. Glycolysis proceeds as previously discussed, ending with 2 pyruvic acid molecules, a net gain of 2 ATP, NADH, and then you convert glucose into 2 molecules of pyruvate or pyruvic acid. Glycolysis is then followed by a fermentation reaction. The fermentation reaction occurs in 1 or 2 steps, depending on which fermentation we're talking about. Fermentation does not require oxygen. It does not use the Krebs cycle or the electron transport chain. It only has glycolysis and then 1 or 2 fermentation steps attached. It uses an organic molecule as the final electron acceptor, such as pyruvic acid, and then the pyruvic acid will be changed into the fermentation end product. To make the fermentation reaction, you use up that NADH molecule, which was made in, come on, mouse. The NADH made in glycolysis is used up in fermentation to precede the fermentation reaction. And then pyruvic acid is converted to the end product of fermentation. There's 2 fermentation reactions I briefly want to talk about. That's lactic acid fermentation, where glycolysis is converted and converts glucose into lactic acid, and the glycolysis makes the ATP. It does make NADH, but that NADH is used by the fermentation reaction to make lactic acid. And this is used all the time in making food. Lactic acid prevents food spoilage. It's why sauerkraut is sour from the lactic acid. And then the sauerkraut can stay even unrefrigerated for a while. You can use lactic acid to convert milk into yogurt. Lactic acid is used in a lot of food like chocolate and let me think about it. Most cheeses like cheddar cheese. And that lactic acid helps the food from spoiling. The other fermentation reaction I want to talk about is alcoholic fermentation, where once again you have glycolysis making ATP and NADH, and then the NADH is used up in the fermentation reaction to convert pyruvate into ethanol. This is a 2-step process. Ethanol is a 2-carbon molecule. Pyruvate is a 3-carbon molecule. And so one of the carbon is given off as CO2. And in the making of beer, that CO2 makes the beer fizzy. In the leavening of bread, like you put yeast in bread and it undergoes alcoholic fermentation, the CO2 is given off, giving the bread the rise, meaning the risen bread, the air pockets in the dough. And in wine, the CO2 is vented to the air, and so it doesn't stay in the wine. But if you were to cap the wine bottle, you would then call it champagne, and the bubbles are the CO2 made in the wine that we call champagne. Your muscle cells can engage in lactic acid fermentation. Whenever the muscle runs out of oxygen, as long as glucose is available, the muscle can fall back on fermentation to make ATP. And then the ATP can be used to allow the muscle to continue working. And the muscle burn will be the lactic acid building up in the muscle. Now, why does the muscle use up this NADH instead of just storing it? Because if you were to store it, eventually the muscle will be given enough oxygen. This NADH could be used in aerobic respiration to generate a lot more ATP, because this NADH actually being used up does not generate any ATP in fermentation. Why does your muscle use up that NADH rather than store it? Anyone have a guess? It's actually shown right here. Come on. The NADH has to be used up so that we can recycle NAD plus to get glycolysis to happen. If you don't get NAD plus and your cell uses up all its NAD plus, you cannot have glycolysis happen. If glycolysis doesn't happen, you produce no ATP. If your muscle doesn't have any ATP, it cannot contract. And we usually see one Olympic athlete in the Olympics where the Olympic athlete is, I don't know, doing their best, trying to get across the finish line or whatever, and they use up all of their ATP. They have no more ATP. They're using up, of course, all the oxygen in their muscle cell, so they're falling back on fermentation. And if they use up all the glucose in the muscle cell will have no more ATP. The Olympic athlete will actually collapse. Their muscles aren't able to work, so their muscles actually stop working and they collapse. The coach will run out in the wintertime and throw a blanket over them. Their heart and the lungs are working like mad to generate, send blood to the muscles so that glucose and oxygen will come to the muscle. And then the Olympic athlete, they're fine. Their muscle cells can then begin working with aerobic respiration, making ATP, and they'll walk up and cross the finish line. In the wintertime, I imagine the coach comes out with a spray bottle and just sprays them down. And then the same story happens. Any question about any of that? All right, there are different fermentation reactions. We've talked about the two main ones, ethanol and lactic acid fermentation. A lot of our fermented foods happens by lactic acid fermentation. Most cheeses, in fact, every cheese except for Swiss cheese that I know of follows lactic acid fermentation. Yogurt, sauerkraut, whatever, any fermentation reaction. Naturally fermented pickles. Yeast can make beer and wine or raise bread, and that follows alcoholic or ethanolic fermentation. Swiss cheese uses propionic acid fermentation, and Swiss cheese has holes in it, but holes are made in Swiss cheese by the making of CO2 and propionic acid fermentation. There's a few other fermentation reactions. We're not going to talk about them. I think it's this one, makes vinegar. They can be used in industry to make various products like isopropyl alcohol, maybe lactic acid. And this is showing you some industrial uses of different types of fermentation. Do not memorize them. It's showing you the fermentation end product, ethanol, lactic acid. Oh, there's rye bread, sauerkraut, summer sausage. Ethanol also used to make fuel for your car. I'm not going to talk about those. So this is a good summary table looking at aerobic respiration, anaerobic respiration, and fermentation, and then comparing them. The growth conditions of aerobic respiration have to occur with oxygen. So the growth is aerobic, where oxygen is present. The final electron acceptor is oxygen. Anaerobic respiration happens anaerobically. The final electron acceptor is a molecule other than oxygen. In fermentation, it's an organic molecule. And fermentation can be either in the presence of oxygen or without oxygen. And then you don't need to know about this one, but ATP being produced for aerobic respiration for prokaryotes, you always get 38 molecules of ATP per molecule of glucose. Eukaryotes, most of their cells are not as efficient, and they generate 36 molecules of ATP per molecule of glucose. Of course, the heart and the liver and the lungs are a little more efficient. And then anaerobic respiration, it's variable, the amount of ATP you make, because there's different forms of anaerobic respiration, and it depends on the form of anaerobic respiration. But it's always less than 38, the highest is 36, and it's always more than 2. And then fermentation, you only get 2 molecules of ATP per molecule of glucose. Any questions about any of that? What we're doing on the time here? Got 1 more minute. Let me briefly state that I've talked about carbohydrate metabolism for generating ATP, and carbohydrates generally will be converted into glucose and then sit down aerobic respiration the normal way. Lipids and proteins can also be catabolized to generate ATP. There's one problem, and that is a lipid and a protein are too large of a molecule. These large molecules cannot enter the cell and cross the cell membrane. So what the cells do is they produce lipases and proteases and then secrete these enzymes extracellular and then digest the lipid and the protein outside the cell, making smaller subunits. With proteins, you make amino acids. With lipids, you make fatty acids or glyceride, is that it? And then you bring these smaller molecules into the cell. And that's the same way our cells digest lipids and proteins. We digest them outside the cell by secreting lipases and proteases. With proteins, and amino acids, the amino group has to be removed from the amino acid, and then the remaining part of the molecule will enter aerobic respiration, generally in the Krebs cycle. I'll show you that next time. And then you'll pee out the ammonia. All right, any questions about anything? If not, we'll continue here next time. All right, if there's no questions, I'm going to end it. I'll see you in the lab. Wait a minute, let me look at that again. I think we have a lab today. Yeah, we have lab 7. All right, I'll see you in the lab at 6:30. Bye.